My title is a play on a movie that played maybe a couple of years ago, Inside Out. How many saw it? Yeah. So Outside In is really us taking in what's there in the world and forming a world in our minds, okay? How do we form this world in our minds? We form it through senses. And this invokes not just the piano's music, but certainly you can kind of imagine music. Now, if our eyes could not see the keys and could not tell the black from the white, okay, we would be hard-pressed to play the piano unless it were by touch alone. So thank God for touch, we'd be able to tell the differences. But note how the piano keys are sort of meant for the size of our fingers, right? I read of some piano greats designing keys to be just a little narrow so they could stretch and play more difficult chords. So the world that we navigate in is a little bit sized to our convenience. As well it should be, no? I mean, Gulliver's Travels, where this giant shows up in the land of little people, it doesn't work, right? Our world is sized to us. But we also delude ourselves into thinking that's all there is out there. We see what we want to see, okay? And you're all thinking we're scientists, we are pursuers of the truth, you know? Of course we know what's out there. Okay, we do to some extent. But let's start with an experiment that will begin to even challenge that. If you all have this piece of paper, just roll it along its shorter length. You want to make a roll like so. Yeah? The hole should be about the size of a, let's see, a lemon, small lemon. Yeah? Like so. Now, hold the roll in your right hand, like so. Yeah? Just hold it so, lightly. Now, your left hand is going to be in two positions, okay? Make it flat, and it's either at the top of the roll or at the bottom of the roll, but never blocking the roll. Yeah? On the side at the top or on the side at the bottom. Okay? One position, two positions. Yeah? Now, keep it at position 1. Keep the left hand at position 1 and just raise it to your eyes and look. Now both eyes are open, okay? Both your eyes have to be open. Your position 1 has to be all the way to the top of the roll so that your palm is pretty much obstructing anything you can see with your left eye. Your right eye is looking through the hole and the left eye is obstructed by the palm. Everybody got that? Can you all see me through this? Half the people aren't doing this. You don't have paper. Please get some paper. Oh, come on, please get some paper. Just take anything and roll it. If you have a full sheet, tear it in half. It works with half a sheet as well. Don't make it a narrow, very short roll, okay? You want the roll to be about 8 inches. On the other axis, roll it on the other. Yeah? If you have half a sheet of paper, roll it to make a short roll, a long roll, sorry. Okay, are we good? Now we have to go through the position 1, position 2 again. Roll in the right hand, left hand, position 1 and position 2. Just practice both positions. Yeah? And keep both eyes open. Nobody's doing squinting stuff here. Keep both eyes open and just raise it to your eyes so that one eye is just blocked by the palm and the other eye is looking through the hole. Can you see me? Can you see me in that lovely little James Bond-like circle where I walk like so, and then I turn and then I Okay, keep it there, keep it there. Okay, now just move the left hand to position 2. Can you see me? What do you see? Okay, these are like undergrads and PhDs from IISc, right? and they're telling me there's a hole in their what do they serve you for lunch? No, I'm just toying with you. Of course you see it. In fact, if you didn't see it, come and talk to me afterwards. I'm a neuroscientist. Okay, how many people see a hole in their palm? How many didn't see it? It's okay. Nothing dramatic will happen to you. Yeah. Okay, so what do you think is happening, right? Is there a hole in the palm? So this is how I open my graduate level neuroscience course. I open with a debate on whether this apparent hole in your palm is real or whether it is a figment of your imagination. And I divide the class into the realers and the imaginers. Okay? So we could do that with this audience. Are there anybody who says the hole in the hand is real? Some brave soul? Depends on the definition of what is real. Okay, because as you're wisely trying to say, the hole in the hand is as real as anything else out there, no? Okay. Real is what you can see. Okay, real is what you can see. So then there is a hole in the hand because seeing is believing, right? Okay, how many are in the seeing is believing category? Just switch and do the other hand. Your eye dominance may be conflicting, just do the other hand and you may see it more really. I'm not kidding, I'm not kidding. Okay, this experiment just mystifies me. It completely mystifies me how our brain puts these images together, right? When the eye is here, one eye is basically blind. When the hand is up here, the eye is blindfolded, not doing anything. When it's here, one eye is seeing the image through the hole and the other is seeing the palm. And the brain does what its circuits have been wired to do. It overlaps information from both eyes to present a rich view of the world. Our circuits grow this way, okay? In fact, our circuits grow this way through visual experience. Okay, a child is born, has visual experience, and the nerves are wiring up to the cerebral cortex to create the circuitry that produces this. Why do we need this circuitry? It gives us depth perception. Okay, because one eye has a perspective, the other eye has a slightly shifted perspective, and depending on how far the object is you're looking at, the shift in perspective is going to be slightly different. Students of art know this. Okay, students of neuroscience learn this sooner or later. In fact, this particular circuitry of ours is taken advantage of to create 3D movies. Okay, where you have the glasses, the red and the green glasses, and the movies are simply the cameras of two perspectives just overlapped, such that, you know, filtered through one eye or the other glass, one pane or the other pane, you will get two different views of the world. And the brain does what it can and overlaps them, and suddenly, you know, you have the shark or the missile, you know, jumping out of the screen at you. Isn't it fantastic? And we sit there and laugh and happily accept, okay, that our circuits are being royally tricked. Okay, we are so happy to live in this illusion. And for movies, you'll say, yeah, that's okay. That's the movie hall. I go there knowing I'm going to be tricked. I get my thrills and I leave the movie hall. And friends, I will ask you to think during my talk whether we are currently in a movie hall and whether we even can leave it. Okay, that's sort of, that's the theme that I'm going to persuade you to perhaps joining me in, or then at least convince me otherwise. So, you know, I can maybe live in the real world that you inhabit. But let's go further. Seeing is believing. Bangalore, beautiful city. Lots of people have flowers in their windows and in their gardens. Okay, maybe we can cut out some of these lights in the front. Is that possible? Or you need it to be able to record video? You need it? Okay, fine. Let video rule. So these flowers, these are normal, ordinary flowers. Anybody want to take a guess as to what color they are? This is not a trick question. Yeah, so I too see purple and white, and I too see yellow. But you know, what color are the flowers? Is there such a thing as the flower having a color? Okay? Because after all, color really depends on who's looking. And here are these flowers photographed in ultraviolet light. Okay, I mean, who would have believed, right? And you can imagine these patterns are perhaps more relevant to, say, bees that are pollinating these flowers, and bees can see in ultraviolet. Now, I'm slightly tricking you because fact is we're the ones that are doing the seeing here. I cannot tell you that this is what the bee sees. Okay, so, but the ultraviolet illumination is bringing out these inherent patterns in the flowers. There is an inherent pattern that is not directly visible to us. Okay, the flowers were not there for us. You know, we take advantage of them, but the flowers are actually there so that flowers may produce more flowers effectively, right? So this is the world seen with a slightly different perspective. And now who's to say what color the flower is? Okay, one has to naturally ask, is to whom? And let's explore a little further. So I clearly see that there is a nice white square. Is there a pointer here somewhere? No? Okay. I clearly see a nice white square card paper laid on top of 4 black discs. Okay, everyone with me? That's sort of, anybody doesn't see it? Nobody's going to dare to say they don't see it. Okay, now I'm going to persuade you that this white square on top of 4 black discs is a lovely illusion that's been created by our circuits that demand for us to make sense of the world. Okay? Presented with this kind of cutout, our circuits are creating this artificial edge here. How many people see a slight contrast between this and this? Yeah? Yeah, you know, it's there, right? Or is it? I'm going to start covering things up one by one. There isn't anything there. There's no contrast there. That edge was completely fabricated. It was an illusion because our circuits are meant to help us make sense of the world. Okay, and these illusions bring out where our circuits actually just make things up. And we're completely cool with it. Here's another. How many see lovely little white circles? Yeah, at each intersection. In fact, I sometimes see highways. Yeah? Again, same thing, okay? These are illusions created because our circuits are the way they are. Why are our circuits the way they are? Because, well, it helps us live in the real world. We'll get to that later. But it's not just edges and things. It's even that which makes our world so rich and delightful. It's even color, okay? Now, I won't push you to guess, but I'll say that, you know, I kind of see a orangey background with a brown dot in the middle and a brown background with an orangey dot in the middle. Can we close the lights for a second to just, just somebody, just close the front lights for a second so that we can, yeah, perfect. Actually, no, not. Is this okay? This fine? Okay, then let's leave it like this, thanks. So, yeah, I mean, I see brown dot, orange background, orange dot, brown background. Do you all? Yes. It's okay, there's nothing wrong with you or me. But now, let's just connect them. Okay, it's actually the same dot. I just cut and pasted a square from here all the way to here. The color is actually the same throughout. Yet even now, my brain is insisting that this half is more orange than that half. Okay, I actually just cut pasted the same square over and over. Okay? So color is highly context dependent. We don't even know what color the things are. And you know this, right? I mean, in the dusk things change, in yellow light things change. Okay? So it's our circuits playing tricks on us. And here's a lovely one. If a computer, not a AI machine learned computer, but just a regular, you know, we're asked to look at this image. Thank you. We're asked to look at this image. What is the most logical description of this image, the most minimalist description of this image? Water bottle? Water bottle is a highly enhanced version of this image. It's actually a little green cap with a plasticky bit, and then a little plasticky bit like this, then a little bit with green and transparent, and then another. It's actually bits of a water bottle. How do you know there's anything behind those black stripes? We're just making it up, right? You don't actually see it. Yeah, but you know, now the amazing thing is the amazing thing is, right, so you can do, psychologists do tests on babies to figure out when various abilities come into play, okay? And how do you get a 3-month-old baby to tell you what it's seeing, right? I'll describe an experiment, not about water bottles, but how babies can be evaluated for math, okay? 3 months, 6 months, 9 months, okay? They're sitting with their mom and babies suck, okay? And one interesting feature about babies is that when they see something interesting, they suck harder. Okay, now this can be used. If you show a baby a screen and a ball rolling behind the screen, then another ball rolling behind the screen, then the screen goes and suddenly there are 5 balls. Okay, the baby's telling you that it's surprised, which means it had an expectation. You can only be surprised if you have an expectation. So there is a certain age at which, okay, a baby will demonstrate the ability to do this kind of math. It's amazing, right? These circuits grow. There is an age at which a baby thinks when something disappears, it's gone from the universe, okay? Which is why ooh, ha ha works. Yeah, because they continuously get surprised. There's a certain age after which it won't work. The baby look at you and say, really? Okay, but it's these lovely baby circuits growing, and they're growing to help said baby survive the world, okay? Because if it didn't, there would be no more babies and the human race would die. So everything we have, all of this richness and this color and everything is essentially so one egg can make more eggs, okay? I mean, it sort of gives us it's a humbling experience, right? I mean, think of it. The purpose of an egg is to make more eggs. And every exam you've taken and heartbreak you've had and every piece of music you've learned and every, you know, experiment that crashed and every fight you've had, every dinner you've, you know, enjoyed, every party, everything you've done is just small blips on the grand plan of one egg to make more eggs. Okay, and necessary in this that said egg or the product of said egg be able to survive. And what is this trait? Okay, this trait is image completion, and it's incredibly necessary to survive in the world because, you know, images are not complete when they're presented to us, right? Images are often incomplete. God forbid if somebody, you know, some early human thought that this was little bits of unconnected tiger. Yeah. We have achieved this skill of completing an image even though all bits of it are not presented to us, okay? It actually helps if the image moves. You can't see something hidden until it moves, and then suddenly you extract a shape from these hidden bits. Okay, that's high-level circuitry at work, and we do it effortlessly. Okay, our circuits are wired that way. My theme is everything that we experience is because our circuits are wired this way. I study how said circuits get wired. Okay, so we've gone elaborated on vision. We're going to now look at sound. It's not just vision that lends itself to being deluded. Sound is a wonderful, wonderful way of deluding. I mean, you all know this, right? Ventriloquism. Forget ventriloquism. I'm right now playing a monstrous joke on you, right? Where is my sound coming from? It's coming from the speakers. Okay, you're not actually hearing me talk directly, you're hearing me through the speakers, and yet, because my mouth is moving, you are willing to ascribe the sound to me. Haven't you been in a large hall where suddenly some question in a thin voice booms out from the speakers and you have no idea where the person is? And then when you finally nail the person, then suddenly your illusion of sound becomes ascribed to that person? That's what ventriloquism is. They just move something in synchrony and they don't move their lips. So then it looks like this is doing the sound. Okay, I practiced that one. Don't know if it worked, but thanks for clocking. Thanks. Okay, I mean, we're just willing to assign sound to anything that's moving in sync. Okay, and we are so persuaded by our vision that we, in fact, see sound.
Ba ba, ba ba, ba ba, ba ba, ba ba, ba ba. ba ba, 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 ba ba.
Now, I'm going to play all three side by side, okay? Because here's the kicker, it was the same soundtrack each time, okay? It was Baba each time. We're going to play that soundtrack and you're going to look at the three faces, and depending on which one you look at, your percept of sound will flip. It's freaky, okay? And if you don't look at all and if you just close your eyes, so for each one you can close your eyes, it will immediately flip to Baba. When you open it, it changes depending on what the lips are saying.
Ba ba, 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 ba ba.
Cool, huh? For how many people did that work? How many people saw sound? Freaky, no? Really freaky. So think about how our brains really want to make sense of the world, okay? So when you hear a sound, you use lip reading skills to try and make it all match. And your brain tells you that this is what it is, okay? So seeing may not be believing, hearing may be seeing, and now let's do touch. Let's go through all the senses. Let's see how good we are. You know, this hole in the hand, sometimes when I do this, my students will tell me, you might be seeing the hole, but touch, no? There's no hole in the hand. So you're using one sense to cross-check the other. To which I ask, as you might, touch with what? What is your probe? What is the size of the probe you're touching with? Okay? I want you all to close your eyes and imagine a tennis net. Or maybe look at one. Okay? Now close your eyes. I've given you a tennis net and use your fingers to probe said material and tell me, does this material have holes? Poke the net with your hands. Poke the net. Does it have holes? Are your fingers going through? Yeah? This material has holes, right? Okay, very good data acquisition. Now go to a mosquito net. Close your eyes and poke and tell me, does this material have holes? If your mosquito net has holes, you're in serious trouble. But all I'm trying to say is, we're sampling the world with the probes at hand, right? If our probes do not have the resolution, you're not going to see aspects of the world. If instead of fingers we used toothpicks, we would be able to conclude the mosquito net has holes. If we set an ant loose in the grass outside, the ant would report that the world is a nightmarish forest everywhere, full of mountains and everything. Okay? If you set an ant loose upon a real mountain, real for us, you think the ant would notice? It's the same bumpy ground, whether for us it's flat and for us it's inclined. For the ant, it's bumpy ground. Okay? Now, how is the world? It really depends on who's looking, at what scale you're looking, with what limited, hopelessly limited sense organs you're looking, right? And we are so full of hubris, pride, that we, the most evolved, we humans with our big brains, we know what's out there. We're so proud. We have nothing. We see in an itty-bitty, visual spectrum. There's wavelengths here and wavelengths there we have no idea about. And we think we know how the world is. Okay, we plunder through, with our big, enormous fingers, and we create things that are the size that our fingers can grasp, and we're like, okay, that's how the world is. It depends on who's looking. We don't even know what color it is. Okay, we don't even know how, what is making the sound. We are led by our eyes. We don't even know whether the material is rough or smooth, because that is, again, a matter of scale, right? We really know nothing. But how sure we are that we know. Being a neuroscientist actually drives this home. If there's anything all of my research brings me to think about, is really how puny we are, how little we are, and we are so full of knowledge. And, it's a sense of apparent knowledge and control on the world. So, students out there, I would say question everything. There's always things that we just don't know. There's things that, maybe we cannot know. And these are philosophical matters to debate, over late night drinks. Okay, but what do we research then? We research, well, how is all this built? How is this circuitry that produces this magical, magical world built? Because, illusion or not, so Hindu philosophers had it right? The world is Maya. Illusion or not, it's one fabulous Maya. It's wonderful to live in this. Is this beginning to sound like the Matrix? It's such an enticing, and this is why great minds have decided to abandon it and, try and achieve something beyond it. For now, we're living in it. So what we look at, what we are fascinated by is how is this circuitry of sensation wired? Because it is really responsible for creating this whole world in our minds. Okay? So how is this, I wish we could grow a brain like this, physically wiring it up. We can't actually go and buy motherboards and, RAM chips and so on, and we have to grow it. We have to grow it from a single cell, which means we have to grow every single component, and having created the components, we have to assemble them using cell migration in the correct places, and then we have to wire it up, and all of this is a biological act. Okay? Are there any people who are not biology majors or biology background? Okay, so welcome to our world. Biology is highly welcoming of people from other streams because it's a giant engineering problem. Okay, look at it. Here I'm going to show you 2 videos if they will play. This one is a cartoon of a human embryo. 1 cells become 2, 4, and so on. That's now embedded in the uterine wall. It's making placenta. This blue thing is going to become the embryo. The rest of it is support system. And watch now the beginnings of the vertebral column come in. Watch how the placental network is nourishing it. Look at the head, the limb bud, the eye, the umbilical cord. Yeah, the vertebra forming. The human baby spends most of its time growing the brain. The rest of the body is kind of stunted. It gets born when the brain is kind of barely there. And here we go. 9 months coming up. And 1 more baby has entered the matrix. Okay, here is a real video of a zebrafish embryo, which does a good bit of this in 1 day. Okay? So ball of cells, it didn't even wait for me to count 2, 4, 8, 16. Ball of cells, that's the big yolk. The cells wrap around the yolk like a mask. And then, before you know it, the neural tube has formed. That's the long axis of the embryo. You'll see an eye coming up here. And while I was distracting you with the eye, the beginnings of what will be the vertebral column have formed there. And now the tail extends. This happens over a few days. Melanocytes come in to color the skin. A nice straight little fish embryo. Almost ready to start swimming. That's amazing, huh? Look at the time index. I mean, this is like 2.5 days. What did you and I do in 2.5 days? This thing created itself, okay, from 1 cell. This is sort of the magic of biology, right? A program on autopilot. I regard an embryo much like sending some, shuttle to, some distant planet with all of the instructions it needs to unpack itself, take raw materials from the environment, and eventually make more of itself. with no communication with anything. I mean, that's really what an embryo pulls off, right? It's pretty much on autopilot, and it creates a whole little individual. Okay, the most interesting thing for a neuroscientist of this individual is its brain. This is a cartoon of a human brain, and just going to show you the basic simplified visual pathway. Haha. This is a simplified visual pathway. I'm not going to walk you through all of this. Just to tell you that this is the connectivity required, right? The nerves from the eye have to connect via a relay center that I'm going to talk about, and then basically, so that's the retina relay center, and connect up to various parts of the brain so that we can do color perception, edge detection, binocular vision, and so that then we can eventually associate sound with what we see, and so on. Okay, here's a piece of cortex that does this. Cerebral cortex, our highest processor, 6 layers of it, they've been kind of color coded, and each little particle you see and each little line, so each little dot is one neuron, and it's got a long input wire called a dendrite and an output wire. Magnified, a neuron can look like this, a sort of triangular pyramid-like shape, and each red dot on this is where some other neuron has come and connected with it. Okay? So on average, each neuron has between 1 and 10,000 connections, and the brain has 100 billion neurons, okay, each with this much connectivity. I do not think this is a challenge for an average electrical engineering problem. Okay, I don't. This is a possible for a, you know, to wire up a car. I mean, if the nano, for example, you know, a nano size wiring in one's head, I think an electrical engineer might just run away screaming in terror if they were asked to do this, right? And the embryo sort of executes it, and executes it with amazing precision. So the components have to be grown and moved in place. I'm going to show you a video. This is a slice of embryonic cerebral cortex from a colleague, Kazunori Nakajima, where each red object is a newborn neuron that's going to migrate. So it's going to translocate itself to its correct position where it needs to be. And look at the activity, okay? I mean, look how each cell seems to know where it's going. Some of them go kind of in directions, but maybe they'll correct themselves, or maybe that's where they're supposed to be. Look at each cell sort of sensing its path and growing, yeah? Pretty amazing, right? And this is happening in the embryo's brain in order for the components to get assembled, okay? After they get assembled, the wiring has to form. So we're now going to look at how does the circuitry of sensation form. This cartoon is a cut through a mouse embryo's brain. We use mouse as our model mammal. So these are the two halves of the brain, so like so. And if I cut through the brain, I'm going to see these two halves, and this sheet on the outside is the cerebral cortex, and the relay center I mentioned is here. Why relay center? Think about all the sensation coming in from your entire skin. Okay, think about the hearing information coming in from the ears, and then the visual from the eye. If all of this were just growing helter-skelter into the brain, it would look like, you know, one of those terrifying, you know, first-year undergrad wiring projects where, you know, connections are made mid-air or something, okay? It has to be systematized. So all of the sensory information, okay, except olfaction, which goes through a different channel, comes to a centralized relay center in the brain, and from there it's parcellated out to the cortex. So that's this, okay? So that's the relay center, and we're going to look at the sensory part of it, the skin sensation, and that's the pathway that goes to the brain. Now, if this were a system where every part of your skin had equal access to your brain, you would have equal resolution with every part of you, okay? Do you need it? Does the back of your ear need to be able to feel as delicately and precisely as the front of your fingertips? What are you going to do, you know, with the back of your ear, right? Okay, or your stomach, for example, or the back of your elbow. You don't need resolution and discrimination in every single part of you, right? And yet, the amount of brain we have is the amount of brain we have. It's like a processor. It's like a, you know, imagine a megapixel, you know, multi-megapixel. What's the best camera? 64? More? Less? So you would buy more and more megapixels if you wanted good resolution, right? But when you do that, every part of the image has that same resolution in it. The brain doesn't do it this wastefully. Rather, it says, well, parts that really need high density representation, proper resolution and discrimination, such as the fingers, such as the lips, kind of important because you have to know what you're eating, those parts will have access to more territory in the cortex, and the others will have less. This is a very intelligent way of designing this, right? So if I show you a map of the body surface scaled in proportion to the amount of processor territory it has, here's what we would look like. Okay? This is actually very clever. Okay? Some parts of our body have access to a whole lot of cortex, and other parts not so much. Okay? Look at the hands, the arms, puny, no? Compared with the fingers. So this is actually the clever way in which the pathway is designed, and so it is in all mammals. Here, for example, is a mouse. For a mouse, its whiskers are like fingers, okay? Whiskers of a mouse aren't just cute things we draw in cartoons. Whiskers are actively whisked, and when a mouse has to go through a gap, it actually can whisk and sense whether the gap is big enough or not. Okay, it actually whisks, and in fact, it's very cool. There's one whisker down below under the chin, or a set of very big whiskers, and when a mouse goes on territory, that whisker is effectively doing atomic force microscopy on the terrain. Yeah, I mean, bump hai, nahi hai, okay? It's actually doing that. The brain's already sorted it out. AFM is just on a different scale, that's all. Correct, right? Physicsy people? Okay. So here's the mouse. In proportion to the cortical territory that its sensory surfaces have. And sure enough, the snout, the upper jaw, the lower jaw, and the whiskers have a lot of territory. The little bit of the back of the leg and so on, not so much. Forepaws, much better than hind paws, because a mouse actually uses its forepaws to feel things. Survival. It's all about one egg making more eggs. Okay? Think about how evolution might have designed this circuitry. A mouse which didn't do this, bias its allocation of territory, is going to feel danger a little less, and it's going to go fall in some puddle or something, right? It's going to be wiped out. So you can see how this strategy might have been evolutionarily selected, and the next gen of the mice and the next gen and the next gen evolved this strategy. Now, whiskers, you see these dots, the ovals, right? Whiskers are not just one random amount of just whiskey hair. Each whisker has a name, and each whisker has a location on a map, which means sensory information from each of these whiskers, okay, gets translated in a nice map from the whiskers to the hindbrain, to the relay center, to the cortex. This is just a high mag view of the cortex, where basically this one whisker, if it was cut out and removed, this one C2 patch of the brain would not show any input. It's a beautiful map. Okay, it's sort of like taking the map of Bengaluru and translocating it, so the up, down, right, left modalities may get moved around, but basically your house is still next to your neighbor's house. This whisker is still next to this whisker, represented in the brain, and that's what's relevant. Okay, this is called a somatotopic map, a sensory and topologically correct map. In fact, each of these things are called barrels. They were named so by the scientists who discovered them. Barrels. Are there really barrels in the brain? Up until now, I've only shown you cartoons. Okay, so as students of science, you should ask to see the real data. Are there really barrels? Well, Woosley and van der Loos, who named them, may have liked beer a lot, and they looked at barrels and barrels of beer. And when they stained up just the surface, flat surface of the cortex for an enzyme called cytochrome oxidase, here's what they got. Okay, so each whisker corresponds to one of these clumps. So that's why they call them barrels. And this is a very easy way for us to tell whether the whisker to brain pathway has formed correctly or not. That's why I'm showing you this. If we take a mouse and stain it up for this, and we see this pattern, okay, so this is the primary whisker field and all these are other parts of the body. If we see this, we know that the pathway has wired correctly. And we look at the whiskers because they are like the major sensory organ of the mouse. Now, my student Ashwin discovered a gene that controls this, okay, and that's some of our research. He found a critical gene that controls the making of barrels. Because if this particular gene was knocked out, removed, we can make a mouse that's mutant for a particular gene. And we didn't remove it all over the body, we removed it only in the cortex. We didn't remove it in the pathway that's actually coming into the cortex. That was normal, but when that pathway enters mutant territory, here's what Ashwin found: hide nor hair of a barrel. And this was unusual because we didn't do anything to the whiskers. We didn't do anything to any step of the pathway. The pathway was completely normal, but when it hits this cortex, suddenly no barrels. So that's actually what we're currently working on, and I'm going to give you a little glimpse of where the project is at. So this now is a real chunk of brain, okay, 2 hemispheres, and here's where we placed dye that has the ability to track along the nerve, so we can see where the pathway is at, where did it get stuck, right? That would be a legitimate question. So here's a cartoon, for example, if I show you this half, we place the dye there and we're going to ask, well, where did it get stuck? And what we're going to see is sort of that's where the dye placement and that's the pathway growing in. Isn't it beautiful? These thin fibers growing into this is going to be the cortex. And now, here's normal. Here's the pathway come in, and you can see the pathway has come into the cortex, and you can see these barrels, right? You can see clumps. And here's the mutant. The pathway has come in, but no barrels. Here's high mags of the same thing. Barrel, barrel, no barrel. So we now can conclude that there is something that the pathway enters okay, but at the point of entry, something goes awry. So this is how you move the question forward. Where is the problem? My student Suranjana, so we don't actually know, I'm just going to tell you where we are at. For this, we're going to look at something called the subplate, which is best described as a park and hold array for these incoming nerves. The incoming nerves come in and in the mouse just wait under the cortex for 5 days before they actually go in and make barrels. Why this waiting period? Not entirely clear. Okay, it's likely that they wait because the rest of the cortex has to assemble. What they do with the subplate and what they get from the subplate is also not clear. But, so here's the cartoon describing it. So if these blue dots are the subplate, then these fibers come in and pause at the subplate before they go in. And my student Suranjana decided to ask, well, is something wrong with the subplate? Okay, so this is now a section, that's the cortex, and this stain is for subplate. It's a molecule that's present in subplate. And this is normal, nicely, tightly packed subplate. And see here, with my X-ray eyes, I can even see kind of barrels, can you? Yeah, so this is the subplate, and here is the mutant. Disordered and poorly formed. So she's already found that when you take out this gene in the cortex, what you disrupt is the subplate. Okay? And it's likely that this is the cause of the incoming nerves getting thrown away. So now we have a handle on asking what exactly the subplate does. Okay? Nobody actually knows what the subplate does, but it's critical for sensory innovation to come in, because remember, no subplate or messed up subplate, nothing enters the cortex, and therefore the animal is visually blind, hearing blind, and touch blind all at the same time. No rich world, no matrix. Okay? So that's where we are at for this project. Now, there is a second key element to experiencing the world. Our senses bring in the information and create this experience for us. But none of these experiences would mean anything if we didn't remember them. Okay? If you do not have memories of that experience, if you do not learn from that experience, if you cannot record it, you'll be living in a continuously repeating, shifting time window, right? 50 dates or 100 dates or whatever, another movie. So we also work on a structure called the hippocampus. Okay, not hippopotamus, hippocampus. Hippopotamus is different. Yes? The hippocampus is the recorder of memories. It's not the place where long-term memories are stored, but it's critical to record new memories. And in fact, it's so important that it's one of the few sites in the brain which continuously makes new neurons even in adulthood. Okay? The old dogma was that once neurons are formed, that's all you have. That's true for many, 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 many parts of the brain, but a couple of privileged sites set aside some stem cells and continue to make new neurons, and the hippocampus is one of these 2 locations. So in fact, right now, you guys are all adding neurons to your hippocampus. Tick, 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 tick. There's all neurons everywhere. But not everybody adds the same number of neurons. Okay? The number of neurons you add is affected by your experience. So here is a standard cage and here is an enriched cage. Standard cage, mice in a regular housing, there's food, there's water. An enriched environment, there are wheels to play and tubes to crawl through, little blocks and things to fiddle around. It's an enriched environment. These mice add some neurons to their hippocampus. These mice not only add more neurons, but the neurons make more branches, more connectivity. If people are familiar with this London taxi driver study, okay, getting a taxi license in London, I think this might have been pre-GPS, required basically acquiring in your head something that they call the knowledge. You had to take an exam, which I don't think even graduate students of science might be able to. To get from A to B, how many right turns and what would be on your left side and what right side and which route would you take? They had to have this map in their minds. Okay, and acquiring this knowledge required a lot of experience, as a result of which the part of the hippocampus that handles this actually grew bigger. Okay, and it grew bigger because of extra connectivity, much, much more wires to achieve this learning and memory. Okay. So the hippocampus is important, its neurons and its connectivity is important to learning and memory. One project in my lab, we've discovered how the number of neurons in the hippocampus is controlled. Okay? So here's a brain stem cell, and normally if a certain control molecule, a gene, a transcription factor, is on, this brain stem cell makes neurons. And if you turn off this gene, then you get instead support cells. For people in the know, astrocytes, glia. Both are essential. You can't just have circuitry without the support cells. Okay, both are essential. But this is called the neuron glia cell fate switch, because this particular gene remains on in the stem cells until neurons are made, and then its levels decline. So naturally you begin to make the support cells. We've shown that basically if you keep this gene permanently on, you keep making neurons. So you can add extra neurons to the mouse's brain that was supposed to be support cells. Okay? A postdoc in my lab, Archana, is actually done this. So here's a normal mouse brain. This is a section now, okay? That's the hippocampus. And here, every green cell is actually a new neuron. Here's a HiMag. Look at these beautiful trees, right? Every green blob is one neuron with its arbors. That shouldn't have been there because we kept this gene permanently on. So she's added extra neurons to the brain. And we're actually asking now, do these extra neurons affect the animal's behavior, the animal's ability to navigate the world, and so on. So that's sort of the boundary of where we are at. Okay? But I'd like to now sort of close with a discussion on what all of this means, right? If our experiences and our memories and what we learn are constructed because of our circuitry, and if new experiences and new learning shapes our circuitry, it means that every difficult task, every tough assignment, every impossible skill we acquire actually gives us different circuitry. Okay, it enhances our brain circuitry. It means every time we take ourselves into a situation where we are challenged or where we can learn something new, it shapes our circuitry. So this then means that the experiences we give ourselves can actually change us, because our behaviors, after all, come from our experiences and our circuitry. So I'd like to sort of close with this neuroscience approach to human behavior and our society and where it's going. I'd like to sort of close with asking ourselves whether we cannot consciously open our minds to experiences that may otherwise be perhaps challenging, maybe perhaps uncomfortable, maybe beliefs we had not considered ever understanding or accepting. Can we not shape our circuits to allow us to be more accepting, to be more tolerant, to be more inclusive as a society. Let's grow our circuits for society. Thank you very much.